All right, welcome everybody to week 22 of our neuropsychiatry didactic series. This one is going to be on probably one of my favorite subjects of all time, catatonia, and maybe quite a relatively unique view of it as a possible movement disorder of motivation. I've given this talk many times and changed it a lot, so I hope that this is a more digestible version. Here's our learning objectives, and let's start with history as usual. So catatonia was first described as a psychomotor syndrome by Carl Kalbum in 1874. He emphasized more its motor signs and viewed it as a motor expression of a variety of underlying conditions like that ranged from medical, neurologic, and psychiatric illnesses, which was pretty close to reality, I think. Um, the psychiatric skew really started with Emil Kraepelin around the early kind of 20th century or so, who incorporated catatonia into what was then known as dementia precox, but is now known as schizophrenia. And yeah, this really, historically laid the grounds of how catatonia has kind of gotten stuck in psychiatry, even though that's, far from reality. So over time, catatonia has definitely been seen as prevalent in neurological and psychiatric illnesses. And by the DSM-5 era, catatonia was reclassified not so much as a type of schizophrenia, but more as a specifier or due to another medical condition. And over the more recent decades, catatonia has been reconceptualized as a network level brain disorder, just like a lot of these other psychiatric conditions that we've discussed. And this specific talk is actually based off a paper that I felt like did a good job at combining a lot of these views that was written by Friccione and Scott Beach, which we'll discuss here. And so we're actually going to skip the cultural slides that we typically talk about, and it's going to be a little bit of a different talk since it's not really a predominantly psychiatric condition. As we've discussed with just about all our lectures right now, large-scale brain networks are basically just disparate nodes in different brain regions that tend to talk to each other for specific functions, and these nodes have a functional connectivity, which this paper describes as rich club connectivity. And so, we're just going to lay some groundwork here before we actually start talking about catatonia. But this interaction between the salience network and how it works as a switch between the off state, which is the default mode network, and the on state, which is the central executive network, is a common theme with psychopathology, known as the triple network model of psychopathology. Just to remind us that each of these specific networks has these specific nodes with specific functions, with the default mode network, being involved in the self-narrative function, the executive network, really involved in performing a task, and the salience network as acting as a switch between the default and the executive network, in a sense that's usually driven by dopamine. And acting as a switch to direct our attention, whether it's inward, which is default, or outward, which is the executive. Um, and so a breakdown of these connections between these networks is really what's thought to underlie catatonia. And as we spoke with our previous lecture on OCD, there's these corticostriatal thalamocortical loops that actually are these dense white matter tracks that connect different levels of the brain and help them talk to each other. And if you were to disrupt any of these, it can cause an issue. And this connection between an aberrant salience network, which we see in psychosis, and this connectivity of CSTC loops might actually underlie mechanistically why OCD and schizophrenia tend to share some features. And basically, like we've said, a lesion along any of this, the dynamic connections between the salience network, the central executive, the default mode network can manifest in neuropsychiatric disorders, and catatonia being maybe the most neuropsychiatrically striking. Um, Still, talking about some loosely related principles that we've sort of referenced before in other talks, but we're going to really talk about it here conceptually. These are all made by Carl Friston, who's actually the most cited neuroscientist of all time, who conceptualized this idea of a free energy principle, which is the idea that the brain is very energetically unfavorable, and it works to reduce what's called surprise, which are things that are of course unexpected. And so when something's unexpected, it's very energetically unfavorable to deal with it. And so the brain is this predictive processing machine that's always making predictions to try to figure out what's going to happen next so we can plan accordingly. This is an active like hypothesis testing process in that the brain makes these hypotheses, which are these predictions, and it's tested by what's this process known as active inference, which is using sensory stimuli from the environment to try to refine these hypotheses, and then only the things that seem to make the most sense and be neutral for survival are the ones that actually are thought to enter our conscious awareness through the thalamus. And this whole process is very important in a possible mechanism of consciousness and decision making. And so now, with the basic themes out of the way, we can actually talk about catatonia from a network perspective. And like I said before, this is going to be largely based on this one paper by Gregory Friccione and Scott Beach, which is this kind of cingulate base model of catatonia with some implications for treatment. And so, first, as we always do, we're going to talk about some neuroanatomy. And so here's some pertinent neuroanatomy to familiarize ourselves with, starting with the anterior cingulate cortex, which I think is the most neuropsychiatric aspect of the brain. It really dictates the emotional tone of the brain. It's found in the medial surface of the frontal lobe. It's likely, uh, this kind of hub for all three of the networks that we've discussed so far, along with and through being a hub of these networks, it helps be this like center hub integrating emotion, motivation, and action all together, while also trying to balance any conflicting signals that it's getting from lower brain states. And then also using that all together to mediate salience and dictate where the salience network is going to point to. So we can either, initiate some goal-directed behavior with the executive network or reflect with the default mode network. And lesions here can cause all kinds of disorders. Neurologically, we know it can cause disorders of diminished motivation, which include abulia and akinetic mutism, but also apathy. And then going forward, we also have our supplementary motor area, and this is just interior to our motor strip, and it's involved in the kind of planning and sequencing of motor actions and drive internally generated movements. And lesions here are usually known to cause a lack of movement or akinesia. And so the anterior cingulate cortex kind of generates the will or motivation to move. The supplementary motor area is going to cause the planning or sequencing to move. And the basal ganglia is really the motor area of the brain. And but also, so it's very active in cortical motor planning, but also gates limbic motivation as well, acting as a sort of it kind of works with the thalamus to act as a sort of gate keeper. And so we're going to have a slide about the interaction between of how the basal ganglia and the thalamus work together to gate certain signals. But of course, the thalamus integrates sensory information and relays motivational drive signals to the cortex. It's the T and the CSTC loops. And dysfunction here can dampen the initiation of movement. And so we need this entire circuit to be intact for us to actually have regular motivational movement. And there's some, functional imaging studies that actually show that in catatonia we see decreased activation of the SMA and the ACC. But I believe it's really important for us to know that proper movement really requires all of this to work. And so if we were to put the entire body in a very simple graph, we can start off with the fact that we have sensations from the outside world or stimuli. And the stimuli, could be salient. And so the cortex, which would be here, would analyze this information. And if it's salient enough, would direct its attention to be to plan to do something about it. So it would require some kind of action or movement. And so there's a part of planning that movement here in the effector, and then releasing that movement and causing the actual gross movement, which is the motor aspect here. And so if you were to think of a lesion just on top of this analyzer effector function here, this would lead to what we would imagine would be either dysfunctional motivation, or it would lead to dysfunctional motivation by affecting the circuits connecting to the anterior cingulate cortex. And that in itself would lead to a sort of distorted or ambivalent movement that can look, different in different cases, which we're going to talk about a little more specifically. And so, I know we've hammered the fact that the Sealands network is extremely important, but let's talk about it just from the perspective of motivational control. And so, of course, as per the name, salient stimuli basically gets the salient network to act. And we have these loops that are kind of connecting it to the default mode network and the central executive network through the anterior cingulate cortex as acting as a central hub. And basically, it can only go one way at a time. And if the cingulate cortex is a central hub, and like we said, it integrates, emotional information and energizes action selection using motivation, hence from the perspective of dopamine, which we're going to see in 2 or 3 slides. It actually also talks to the medial cingulate cortex, which is more involved with cognitive control. And so, if the anterior cingulate cortex is involved in more effective or emotional motivation and control, the medial cingulate cortex is more involved in cognitive planning. And so, lesions, with any of this is going to lead to some, odd motivation. But I just want to take a second to talk about how the medial cingulate cortex is also involved in maintaining cognitive control of planned movement. And so the way that the medial cingulate cortex works, it's been proposed to work through this what's called the actor-critic model. And HRL is hierarchical reinforcement learning. And so hierarchical reinforcement learning basically involves sequencing small movements together to be able to accomplish a task. And the way that it does this is through certain predictive models that it runs through under basically below our conscious awareness. And this is done with the interaction between dopamine from salient stimuli triggering involvement of cortical striatal or CSTC tracks in the anterior cingulate cortex, and then basically going into the medial cingulate cortex to try to see how we're going to do what we want to do by tapping into all the possibilities of things that we can do that is in reaction to the predictions that the brain is always making. And so the way that it does this under our conscious awareness is kind of interesting. Again, it's called the actor-critic model. So the actor here is actually um, the dorsolateral prefrontal cortex, which is involved in executive movement via the motor system. That it also involves the SMA, the supplementary motor area, and the primary motor area. So the DLPFC basically interacts with the supplementary and primary motor area to try to go through certain actions or movements and rehearse them. And then as this rehearsal is the actor part. Since there's a lot of different predictive things that could happen, it's rehearsing quite a lot of different predictive things that could happen. And so, how does it choose which movement or action it's going to go with? Actually going to be what the critic decides, and the critic here is the orbitofrontal cortex. And the orbital cortex is really important in weighing out basically decision making and predicting reward and avoiding punishment. And it's thought to work at this level by basically weeding out the actions that are not predicted to lead to reward or lead to the avoidance of punishment. And whatever is most likely, neutral, like it's not whatever action isn't going to kill you or isn't just like obviously wrong based on inputs from, contextual memories that the brain has in the hippocampus or emotional aspects that the brain has in the amygdala then it's going to get like whatever's the most neutral thing that is going to allow the medial cingulate cortex to be like, okay, this can go back to the anterior cingulate cortex, and this can be motivated to happen. And so it's kind of interesting that at this level, yes, the anterior cingulate cortex is a hub. But the medial cingulate cortex actually is a quick segue into trying to find out what we're going to do before the anterior cingulate cortex comes back and actually motivates you to do that thing. Um, and all of that is really motivated by dopamine and really motivated by a calculated value based on different types of dopamine release. And I know we've talked about this in previous talks, but dopamine, especially in our addiction talk, but dopamine isn't really just a pleasure or reward molecule. It's really a learning molecule, and and its pattern of release is very important. So dopamine has a regular tone of release of how much it's releasing at a time. But then on top of that basal tone, there's also the phasic release. So it can, depending on salient stimuli in our environment or even in our thoughts, we can get a temporary reward that will drive the salience network towards doing something. And so if you get, the more phasic dopamine spikes you get, the better the predictive value will be. And so things with high predictive value that's calculated by the anterior cingulate cortex will then go on to motivate the dorsolateral prefrontal cortex to exert strong top-down control into a movement or of course, action or behavior, while things that are just closer to the regular tonic release that dopamine is usually making at baseline is going to have low predictive value. And so this is going to basically not lead to motivational movement at all. And as we'll see, if there's some conflict here, and there's no cortical movement that happens, basically the basal ganglia can actually take over, and we can have basal ganglia-ish type movements. This is cutting edge stuff that is probably largely conceptual at this point, and definitely requires more research, but. Basal ganglia type movements look pretty odd, and they look suspiciously similar to catatonia. Um, so This paper is really, really complicated. It's not only this paper, but there's also like a supplementary paper where this chart is from. Um, and it all comes together. But we really need to talk about movement here. Um, and so whenever we talk about movement, of course, the basal ganglia is really where where it all comes from and what we talk about a lot. But it's also helpful to kind of have in writing here all the parts of the basal ganglia because there's a lot. So we've mentioned a lot of these things, in different talks, especially the one in addiction. Places like ventral striatum, the ventral pallidum, of course, the nucleus accumbens, the ventral tegmental area. These are all important, but they're just one part of a very large, very complicated structure that in med school we tend to learn more like this. And so this is a functional way of trying to understand the pathways in the basal ganglia. And here we have three pathways that determine movement. Starting from to the far left, we have the direct pathway which is mediated by D1 dopamine receptors. And this pathway leads to basically, it's called the go pathway because it allows for movement. Then we have the indirect pathway, which are D2 receptors. And this leads to stopping movement. That's why it's called the no-go pathway. And then we actually have a hyper-direct pathway that we don't really have to talk much about right now, but it also, I believe, lead to movement. And so each of these pathways are defined by the type of dopamine receptors that drive them. I guess just as a reminder too, D1 these are D1 type receptors. So and they're different types of dopamine receptors. So D1 and D5 are similar and activating. D2, 3, 4 are the inhibitory ones that are also similar in their own ways. And then in this chart, we don't have to really remember it. Um, but the green is going to be glutamate, and the red is GABA. What happens at each level of the basal ganglia here is really complicated. There's a lot of inhibition and disinhibition going on that's going to be beyond the scope of this lecture. But what is the most important part here is basically the arrow, the net arrow that reaches the thalamus. And so what's the thalamus is basically the area of the brain that kind of integrates all these signals into something. And if the thalamus is getting more go signal, more stimulatory signals from the let's say from the direct pathway, then the thalamus is going to basically allow for movement. And so we, conceptually call that like kind of opening the thalamic filter so signal can get through and allow for movement. And then the opposite of that is if the balance is more towards the indirect pathway and being higher, then the thalamus is going to be more narrow and close and not allow for movement. And so the signal does not get through and the movement does not happen. And so, what happens normally is that the thalamus either allows for movement or doesn't. And this is a very sensitive process. And as you see, there's a lot of glutamate and GABA that's involved in this and dopamine given these receptors. So a proper balance of glutamate, GABA, and dopamine is crucial for a well-functioning thalamic filter. And looking at catatonia from a purely mechanistic standpoint, we have very obvious cases of these chemicals not being balanced. As we know, whenever if you've ever seen a catatonic patient, we do an out-of-band challenge. That's GABAergic, and that somehow helps this balance. NMDA stimulation from glutamate is thought to mediate psychosis to some extent, and that could be related to catatonia, and it might be related to at this level as well. And then, of course, NMS or neuroleptic malignant syndrome is a type of basically it could be an iatrogenic catatonia that basically we give somebody an antipsychotic and we actually mess up this pathway in a way that can actually cause malignant catatonia, which is catatonia with autonomic features. And so we'll talk more about the types of catatonia right now. And so going back to thinking about this thalamic filter and whether it's narrow or wide, we can kind of, in a very simplistic way, uh, actually see how different types of catatonia are related. And so, if we've ever seen the types of catatonia, they're pretty striking. The most common is the stuporous or hypoactive type of catatonia. Less common is excited or hyperactive catatonia. And the one that's a little odd and could be life-threatening and needs to be, basically these people would be seen in like in the ICU would be malignant catatonia, which is basically catatonia with the involvement of autonomic instability, which is thought to be basically because of the involvement. One of the ideas is that it's more likely the involvement of the insula because the right anterior insula is important in activating the salience network. Um, and if that, but it also has connections with the autonomic nervous system, with the hypothalamus and the periaqueductal gray. And if it backs up and is aberrant, it can also lead to autonomic instability and malignant catatonia. But anyway, that's aside. What we really want to contrast here is how catatonia can look like a lack of movement, which is the stuporous type, versus too much movement, which is the excited type. And so if you have an excess amount of direct pathway stimulation and decreased amount of cortical control because you don't have the motivational aspect of cortical control, which is usually dictated by the anterior cingulate cortex, then the net result of that is that the basal ganglia signal is going to be what overpowers and what actually eventually leads to the movement or lack of movement. And if it's too much direct stimulation, like we said, that's going to widen the thalamic filter and allow for the release of motor movement. And that's the phenotype of that is an excited catatonia or excess movement. The flip side of that is if you have a relative excess of indirect pathway stimulation. This is going to lead to a narrowing of the thalamic filter, a decrease in movement, and a stuporous subtype of catatonia. And so all of that, that's why something that looks so dramatic, is dramatically different, um, uh, it can still be a type of catatonia. And just really quickly too, these this balance of glutamate, GABA, and dopamine is really important to mention really briefly here. Like we've said before, D2 antagonists, aka antipsychotics, can cause NMS. And then GABA you can actually help with in the treatment of catatonia most commonly, and it's thought to work sort of because it stimulates GABA A receptors and these spiny neurons here, these medium spiny neurons. Um, Along the direct pathway and basically allow for movement to be allowed to go through. And then a lot of neurologic conditions that can also cause catatonia, like TBIs, hypoxic ischemic encephalopathy, prolonged seizures, and all of these different types of conditions can all increase oxidative stress which increases neuroinflammation, glutamatergic excitotoxicity, and can cause GABA shifts and really disrupt this balance and increase the risk of developing catatonia. Um, so here's some, just some, it's kind of interesting to parallel features between catatonia and just what we normally see in neurological conditions. And so, I'm not really sure why they overlap, but it's just, it's definitely there. So, we got to talk about it. In akinetic mutism, it can really look like catatonia. That's even so. As we know, disorders of diminished motivation can range from apathy to abulia to akinetic mutism at the most severe. Severe abulia to akinetic mutism, which this is all a spectrum. The biggest differential of that it would be catatonia. And you can still see some waxy flexibility sometimes, some mutism and negativism with this. And we'll talk more about that in just a little bit. In autism, there's definitely a huge overlap between autism and catatonia, and there might be a component of underdiagnosed catatonia in patients with autism. And you can see this with, sometimes people with autism are mute. They might repeat your words. They might have very specific mannerisms and stereotypies that are important. And Andrew Francis, who's a massive name in the catatonia field, and he's the Francis and the Bush-Francis scale, has actually recently been very interested in the comorbidity between catatonia and autism. There's also an interesting comorbidity between autism and schizophrenia as well too, possibly due to a salience malfunction, but we're not really sure. And then, just about the rest of all of neurology can have some catatonic features. Yeah, and then in schizophrenia, 10 to 25% of people with schizophrenia have catatonia at some point, depending on the study that you see, although it's really, really hard to get a good number. And this might be due to the shared mechanism of aberrant salience. And as we know, the common node involved in just about all the networks, which we'll see really, really quickly, is this right here, which is the anterior cingulate cortex. All three of these networks involve the anterior cingulate cortex, and so it basically, the anterior cingulate cortex kind of works as the bridge connecting these networks together and driving, goal-directed movement with motivation. That might be an association there. And here's just for your knowledge, you can screenshot this or pause the video. But these are some other similarities between many different diagnoses and catatonia. And so Andrew Francis gave a talk on to Bellevue on catatonia and his work with the Bush-Francis scale, and I used his color coding on which parts of the scale are things that we can just get just from observing, which is in red, versus actively, eliciting, which is in blue, or that we actually have to sort of touch the patient and examine ourselves, which is in green. And the Bush-Francis catatonia rating scale, also known as just the BFCRS, is the main way that we quantify the severity of one's catatonia. It doesn't really have a clear cutoff, but on average, just about everybody nobody really ends up with a zero. So I have an arbitrary cutoff point of at least 8 that I use for myself, but I actually don't know where that comes from. But yeah, just know that if you do the BFCRS to a newborn, the number is gonna be not 0. And so I personally just like to break this down into motor symptoms and behavioral symptoms. Again, most of the behaviors are going to be in red because these are just you can just observe somebody's behavior. The one exception is echopraxia or echolalia, which is when the patient mimics you. I wish we had time to actually go through each of these, but we really don't. But echopraxia is when the patient mimics your movement. Echolalia is when they mimic your speech. While mutism speaks for itself, staring speaks for itself. Grimacing is maintaining an odd facial expression. Mannerisms are going to be odd but purposeful movements. And verbigeration is going to be when they repeat the same phrase, like the same phrase. So that's like verbigeration. Or they could repeat the whole sentence, but they just keep repeating the same sentence. Withdrawal is when they refuse to eat or drink, which could be, obviously medically not good. Um, and then some of the most, common features that we see with catatonia is this very specific catatonic tone, which is just provides some initial resistance that almost feels like a waxy lamp, which is called waxy flexibility. And then if they have a stuporous subtype, they're going to be really hypoactive, immobile, and minimally responsive to stimuli, even noxious. They might, if they do withdraw, they might withdraw very, very like abnormally slow. And they might even go anti-gravity and kind of stay there. If they stay there, or if you reposition them and they just stay there, then that's catalepsy. Posturing, stereotypies are going to be repetitive, goal direct or non-goal directed movements, which we of course see in autism very commonly too. There might be some rigidity, and they can have Gegenhalten, which is, or they can have facilitatory peritonia or oppositional peritonia, which almost feels like a volitional. Either you barely move them and they keep moving even more. That's facilitatory, or the more that you move them, the more that they try to resist, and that's oppositional peritonia. Or they might just be extremely hyperactive if they have an excited subtype of catatonia. And each of these can be scored in severity between 0 to 3. And all of these, the Bush-Francis scale is on MD Calc and should definitely be used if you ever suspect catatonia. And so I want to think of this is maybe controversial territory, but I like to think of catatonia under the umbrella of movement disorders, specifically a type of kind of neuropsychiatric or motivational movement disorder. And they have subtypes like stuporous, excited, malignant, and or iatrogenic, which is NMS. And it could have other type disorders that are similar to it, like akinetic mutism, or maybe even severe bulia, and a lot of other neurologic conditions that share features. And so, I'll kind of go further to compare and contrast catatonia with Parkinson's disease in our in one of our neurological lectures on movement disorders. But for now, I'm just leaving this slide here as food for thought. If we can consider catatonia as a motivational movement disorder. And so in terms of its management, first line is going to be out of your run-of-the-mill Ativan challenge or lorazepam challenge, which what we usually do is if you suspect somebody that has catatonia, depending on the studies you see, 60 to 80% of them will basically respond somewhat to at least 2 milligrams of IV Ativan initially. I wouldn't, the same way that we shouldn't be shy about giving Ativan to people with seizures. We also shouldn't be shy to give it to people who we suspect to have catatonia, because treatment is treatment. And a lot of people need a lot more, even at least, 6 to 8 milligrams daily in divided doses. You can give them every, 6 to 8 hours or so for at least 2 to 3 days. And if that doesn't work, then we should very quickly move the ball to ECT. ECT is really it should be the gold standard for catatonia treatment, but it's not because, of course, you have to go under general anesthesia. It's logistically difficult. You have to wait for the ECT team to evaluate them and start them. The times that this is actually definitely indicated is if there's any malignant subtype of catatonia that's suspected. So if there's any autonomic instability, then you should definitely still do the Ativan, but like move to the ECT as soon as humanly possible. And you might actually not get a response until getting at least 6 treatments sometimes, but but 2 or 3 might also be enough to show a response. And so, it's common that people still, have a little bit of catatonia. They're not completely better. And so NMDA antagonists can be used too. Sometimes like amantadine, but that's running the risk of, the theoretical risk of worsening psychosis if that's an issue. And then also memantine is another NMDA antagonist that's a little better tolerated in terms of its psychosis risk, which isn't much. And One thing that I should have said at first is, while we're doing this, catatonia isn't really a primary condition by itself. It's usually secondary to something else, whether it's a structural or functional lesion in the brain. Or and if it's structural, usually we're seeing this in the neuro side of things, and if it's functional, we're seeing it more in the psych side of things. But either way, we should really be targeting the primary condition that's causing the catatonia, in conjunction with treating the catatonia itself. And so if the catatonia, despite all of these first three treatments aren't working, then some other options that aren't as commonly used are anti-seizure meds, like Depakote or carbamazepine, and then or atypical antipsychotics if it's safe to do so, and we're not suspecting a malignant or neuroleptic malignant syndrome as being the cause of the catatonia. And then one thing that I didn't mention here because it's kind of weird and it's really only case studies is a subset of people actually respond to zolpidem or Ambien. But yeah, that you can look into that yourself, but it's typically not very commonly used. We usually stick with like higher doses of Ativan, benzos, ECT, and things like that. And so to put it all together and to summarize what we've talked about, the cingulate cortex is really, especially the interior cingulate cortex, it's really an anatomic bridge that links frontal cortical circuits to subcortical limbic regions. It plays a huge central role in the pathogenesis of catatonia. And this is likely due to it being a central bridge and hub between motivation from the salience network and how it directs attention with the other two networks, the Central Executive Network and the Default Mode Network, and how it coordinates movement from the basal ganglia through the thalamus, and how the thalamus basically allows for that movement to either pass too much for excited catatonia or not enough for the stuporous subtype. Decision making in the back, especially the things that are going on unconsciously, are really interesting from a computational psychiatry standpoint. This is catatonia, in my view, is the ultimate neuropsychiatric condition because this is when we see the functional circuitry of psychiatry and the structural nodes of neurology really coalesce in a way that we can see, immediate treatment response and reproducible signs on exam that either show response or show severity of illness. And so, we really should have a greater awareness of this condition. It can present in all kinds of weird ways, and we can really help people in ways that we know we really can. So we should definitely treat people who have catatonia. And so these are my references. There's also this special article that I pulled some of my slides from too. That is just an updated way of treating catatonia that also involves some of the work that Friccioni and Beach also did as well. So thank you very much. And as always, I have review questions in the PowerPoint slide.